Well, good afternoon, everyone. I hope you can hear me well, and welcome to the fifth of our webinars, GRIP webinars. The title of this afternoon's webinar is In-vitro Biopharmaceutic Methods in the Development of Oral Dosage Forms for Children, which will be presented by Hannah Batchelor, and I'll introduce Hannah very shortly. Just some housekeeping rules, and you'll know that if you've joined us in the past that we don't use video, and we have your microphones switched off so that we don't overload bandwidth for those of you that may have problems. Hannah will take questions after her webinar, and what we would like you to do is to type them into the chat box on the bottom left of your screen. You can do this as the webinar progresses, or when we get to questions. And depending on how much time we have, we'll select some to be answered by Hannah live this afternoon, and others we will put onto the GRIP blog. So GRIP, just for those of you that haven't joined us before, stands for Global Research in Pediatrics. It's a European Union funded network of excellence. You can see the work packages listed here on the slides, majoring on the teaching of pediatric clinical pharmacology with a work stream on pediatric formulations. Please go to our website and find out what we're doing. We'll make sure that you have these web addresses available to you, both later in this webinar, but also in emails that we will send to you when the webinar is over. You'll find then that you can get a lot more information about what we're doing in relation to pediatric formulations and the GRIP project. We're also, as part of the project, compiling an inventory of experts globally, and if you consider you have expertise in pediatric formulations, then please make contact with us. Again, you can email us at the address that we've shown here. So without further ado, I'd like to introduce you to Dr. Hannah Batchelor from the University of Birmingham, who's going to talk to us about a biopharmaceutic methodology and its use in the development of oral dosage forms for children. Hannah's CV has been circulated to you and is on the website, so I'm not going to go into great detail here. But currently, Hannah is working for the Medicines for Children Research Network in the UK, based in Birmingham, where she's a formulations fellow. In a previous life, she's been coordinating and promoting research. She's working on the design and the manipulation of medicines to create age-appropriate dosage forms, and she has a particular interest in optimizing formulations and maximizing their biopharmaceutical performance. So without further ado, I'll ask Hannah to take over the slides, and thank you very much, Hannah, for your presentation this afternoon.
Thanks very much for the introduction. I hope you can all hear me okay. As Tony said, I'm going to talk about the biopharmaceutics methods and development of oral dosage forms for children today, and I work jointly for the MCRN and the University of Birmingham. I wanted to start just by defining biopharmaceutics so that everyone's aware of what I'm going to be talking about today, to make it very clear. Really, on the slide there, we've got images of the dosage forms as well as the gastrointestinal tract here. And what I'm looking at in terms of biopharmaceutics is how those two interact and result in the pharmacokinetic profile that you see on the right. Really it's about aspects of the medicine formulation as well as the gastrointestinal tract. So it's the interplay of these two, and that's what I'm going to talk about in a bit more detail. What I wanted to say here as well is that paediatric biopharmaceutics currently is quite a under-researched area. I tried to do some searches as to what papers were out there, and there was really 4 major papers that have looked at the pharmaceutical aspects of biopharmaceutics. There's several more looking at pharmacokinetic studies, formulations, etc. But the ones that bring them together are these 4 papers here. There was a key paper written by the US Pediatric Formulation Initiative with some key players in biopharmaceutics for those who are very interested in that field. And there was also a paper that was recently put out by a group of us working together for the European Pediatric Formulation Initiative, whereby we reviewed what biopharmaceutics tools were out there and how they could be adapted to paediatrics. It's really going to be those ones that I'm looking at and focusing this presentation on today, as well as some extra bits of information to bring it all to life, I think. I put a so what slide up because really I was thinking about biopharmaceutics, and there's a lot of science in the background, but when I was talking about the pharmacokinetic profile from the outputs, this is really the so what. So the image on the left really compares 2 different formulations, so a paediatric chewable tablet in the blue compared to an adult tablet in the red. And this is a direct comparison of the two products in the same population, showing that by changing the formulation, you've got a difference in the pharmacokinetics. And that goes back to the first slide, that there's product-based differences in biopharmaceutics. And then on the right, shows some difference in the pharmacokinetic profiles as a result of the age. And that's all really to do with absorption, distribution, etc. in the age of the child that we're going to be looking at. This is going from very young to sort of older children. Some of the key tools that are used in biopharmaceutics are permeability. We're talking about drug absorption and how drugs are distributed. So in terms of how a drug is absorbed, it's really to do with how it permeates or crosses a membrane. In this slide here, it's saying that really a highly permeable drug is one where more than 90% of the fraction of the drug is absorbed. For the purposes of today, I'm going to be talking really about oral biopharmaceutics, just really to do with the time limitations. But there are other aspects and different routes of administration that need to be considered. So permeability here, we're looking at intestinal permeability really, and that can be linked to CACO2 measures, which is an in-vitro cell line that can be used to mimic intestinal permeability, or you can use more simplistic chemical measures to do with the drug substance molecule related to its log P, log D, or C log D, and all these things have got correlations to do with the permeability of a compound. A second key measure is the solubility, and a little bit of the text is blocked on this slide, but it's saying the largest dose or dose unit that's soluble in 250 millilitres of liquid, and that 250 millilitres is to represent the glass of water that's taken with their dosage form during a clinical study, and also it's to do with the amount of water that's within the gastrointestinal tract of the adults. Sorry. The third measure there is dissolution, and that's to do with how fast the drug substance dissolves from its dosage form. And a fast dissolving formulation is one where 85% of the labelled amount of the drug substance dissolves within either 15 or 30 minutes, depending on the regulatory guidance that you read. And that's within 900 millilitres of media across a pH range that's representative of the intestinal fluid. as you can see by the big red stamp on this slide, all these key parameters have been fixed and developed with adults in mind. And although it says based on adult anatomy and physiology, they're not exactly related to adult anatomy and physiology, but there are in vitro, in vivo correlations between these key measurements and what happens in humans, in adult humans I should say, sorry. I'm going to go into just a bit more detail on permeability now. So that was one of the key measures used in biopharmaceutics. And really this slide is to look at how that might change with age, see what the differences are in paediatrics compared to in adults or older patients. There are references in the literature that state that the membrane, so the gastrointestinal membrane, is leakier in very young patients. And this is really linked to the phase of development. Newborns are going through a huge growth phase where one of their main functions, I suppose, is to absorb the nutrients for them to grow and develop. Although there's not the clearest of data on this, people have looked at it in terms of sugar absorption, but obviously very little work's been done on drug absorption. The cartoon on the top right of the slide is really looking at drug transfer going through a cylindrical tube to represent the intestinal tract, and on the edges of that tube there are transporter proteins that can either assist or inhibit in the absorption of drugs. You can get active transport or efflux of a drug, and the ages at which those appear and develop is yet to be properly characterised. So that the difference in their presence can really affect the absorption of drugs, as well as subsequently things like drug-drug interactions and the like at those sites of absorption. The table at the bottom really sort of suggests what you might consider to take as being sensible in different age groups. So in an adult, the references for highly permeable data were on the first slide, saying more than 90% absorbed in a human clinical study. Or when you're using predictive methods, things like CACO2 cell lines or chemical measures, you really look at a drug that you know is already highly absorbed and see whether the values are the same or higher than those in terms of its permeability. So you can use adult reference values really from children aged about 2 and upwards, because the data so far, although it's limited, is stating that the efflux transporters are reaching adult values at 2 years. So it's children younger than 2 years where we need to be very cautious about directly extrapolating some permeability data from adults into children. So that's really just to go through what's happening in permeability and how we adapt that for age. Solubility and dissolution media. So when I was talking about the 3 key measures in biopharmaceutics, I talked about solubility, as in how much drug is soluble at the site, and dissolution is how rapidly that solubility is achieved, if you like, how quickly something dissolves. And the key thing that I'm going to talk about here is the media that's used to do those solubility or dissolution experiments. So remember here I'm talking about oral administration, so we're talking about gastrointestinal fluids. So what I've got on this slide here is adult gastric pH is measured quite a few times. It sort of varies between 1 and 2, and adult intestinal pH varies between 4.5 to about 7.5 at the sites of absorption that we're interested in. Paediatrics, on the other hand, there's a lot of variability in neonatal gastric pH about babies being born with a neutral stomach environment, and the time it takes for that to become acidic varies according to the literature that you read, the child's diet, etc. There's a big variability in the pH for the stomach of a neonate. And superimposed on those pH values are a solubility profile for either a weak acid or a weak base, just to reinforce that the drug substance and how it's dissolved at each of these sites can be hugely influenced by the pH at that site. So for a weak base, it's soluble at low pH and poorly soluble at high pH. Solubility will govern how quickly a product dissolves, On the other hand, for something to be permeable, it's a ionized substance. It's more tricky to cross a membrane. So there's a balance between the amount that's soluble and what's not ionized in terms of overall absorption. And the graph on the right is a very old reference from 1953 that shows penicillin pharmacokinetics, and the difference going from the preterm neonate who get the highest Cmax and exposure to a child aged 2 to 13 has been attributed to these differences in pH for that drug absorption. So an understanding of the pH at the site and how that affects drug solubility and dissolution is really critical. So there may be justifications for broader pH range in paediatric populations, or at least understanding which drugs that could be important for in terms of those that have different solubilities over a pH range. Other than pH, there's differences in the composition of gastrointestinal fluids. Typical fluids used to measure solubility and dissolution can range from very simple buffers that really mimic the pH as per the previous slide. These become more complicated as fats, bile salts and other components are introduced to better mimic the actual realistic composition.
Hannah, this is Tony Nunn. We've lost your sound. Can you let us know what's happening if you can hear me? Perhaps by typing into the chat room. Okay, could you help me try to sort out this sound problem by indicating by ticking the green box if you can hear me, please? Okay, we're still having a problem with Hannah, who seems to have been disconnected from us. We're trying to get her back just now. Hannah, I've got you down as Hannah number 2 coming up on the participants list. Can you send us a message? I'm just going to try to sort out your access. Hannah, try speaking now if you would, please.
Hi, sorry about that. I don't know what happened. It just locked me out. Can you hear me?
We can hear you. I may have to change the slides for you, so just do the usual thing of next slide, please.
Okay. Yeah. So on this slide, I was talking about the difference in the composition of the media, and the graph at the bottom shows how drug solubility changes with the addition of the extra components to the buffer. So the addition of bile salts and fats can change the solubility of drugs quite significantly, as you can see in the graph at the bottom. But this is all based on adult data, and there are references that state that there's lower bile concentrations in neonates compared to adults, which could affect the solubility, and also the ratio and the composition of the bile salts in children differs to adults. So the way in which drug solubility is affected by the composition within the intestinal fluid is dependent on that composition, and that's what's poorly understood currently in children. If you go to the next slide.
Hannah, I think you have moderator access now. Just try, please.
I think I can do it now. There we go. So, yeah, I've moved them on now. I think I can do that. If everyone's seeing the solubility and dissolution media volume slide. This is to do with the volume that's present. And I talked a little bit about volume in terms of solubility before, which I'm going to come back to. This slide here really links to the volume of gastric fluid in children. There's various measures of how much fluid there is in the stomach. This slide came up from a reference that it was about 0.56 millilitres per kilogram, which equated to a volume of 37 millilitres in adults, which is quite near to the reference figures, which suggest 40 millilitres in adults. So that just shows you as a child develops the volume of their gastric fluid in the fasted state. I'd like to point that out as well. And solubility and volume used, this graph here brings together some data. The red squares on this chart correlate the 250 millilitres used in adults to the gastric volumes I just presented. So they're just a direct ratio of 250 ml for the different ages. Taking that as one way, and it's only one way, of predicting what the difference in volume in intestinal fluid between adults and younger patients may be. The green triangles come from a reference that Gordon Amadon, who is one of the most significant people in the field of biopharmaceutics, suggested in a meeting in the first publication by the US Pediatric Formulation Act that I highlighted, saying that a volume of 25 millilitres is important for children.
Hannah, you've had problems again, and I think you're back with us now, and you should be able to continue as a moderator, please. Hannah, can you indicate if you can hear me? And if you're back with us, you should have moderator privileges, and you should be able to operate the slides. Hi, it's Tony Nunn again. I'm sorry, we're having problems with Hannah's connection to us. She's trying to get back into us again. If you'll just bear with us for a minute or two longer, I can try to get Hannah back with us. Hannah, if you can hear me, you should have moderator privileges, and you should be able to speak and move the slides on.
Yes.
Oh, Hannah, yes, we can hear you again now.
Yeah, I can hear you. Can you hear me?
I can hear you very well. Do you have any idea what's happening with your connection?
So this is about the solubility and the volumes. Right, good. This is the solubility and the volumes used. No, it just froze, and then it just says that connection lost.
Hannah, at present it's working okay, so I suggest you just keep going. we're not sure what's going on. Barbara, if she's listening, may ask our technical colleagues if they can help us out. If you continue to have problems, we may have to postpone the session, but let's see how you get on.
Okay, I'll try. So this slide here talks about solubility recommendations that we made in the paper, and this is just really a repeat of the slides I've shown, just to bring it all together. Based on the evidence, we were suggesting using 25 ml in children and 250 in adults, and the pH range being questionable at the lower end for younger children, but the same as adults in older children. And then the composition in terms of bile salts, etc., is really hard to predict in the absence of data. The dissolution rate, and where this differs a little bit from the solubility, is to do with the transit times within the gastrointestinal tract. The reason why in adults we say things need to be rapidly dissolving is so that the gastrointestinal transit time does not become rate limiting. So it's 15 minutes in certain regulatory guidances and 30 in other ones. In paediatric populations, gastric transit times are more variable, particularly in neonates. Anyone who's had a newborn realises that the truly fasted state is very limited. They're fed far more frequently, so their intestinal transit times are different because of the influence of the food on intestinal transit. The graph on the right just shows some data of the variability in transit times for different age groups. And a recommendation for dissolution, being conservative, if everything dissolves within 15 minutes, that's the most conservative estimate. So it's appropriate to move that on for children. I've talked to you about biorelevant dissolution, and in dissolution theory people talk about QC, quality control dissolution, and biorelevant as two separate entities. So the volume that I spoke about initially in adult use is 900 millilitres, and that's quite often used in a quality control test. Although we know the volume of liquid in adults is nowhere near 900 millilitres, there's been a lot of papers that do correlate that to what's happening in vivo via an in vivo in vitro correlation. So that's why that volume's been suggested. The volumes in children is a real area of unknown. The pH is there as well as the media, as I said before. And the other thing on this slide to look at is the apparatus. So conventional dissolution apparatus is USP 1 or 2 used in adults. And the reason USP 4 is stated there for the younger children is because USP 4 can work with smaller volumes. It's a flow-through dissolution apparatus, so that you can control the volume to be much, much smaller as you like. And the agitation rate, again, this is a real unknown, and how that correlates to motion within the intestinal tract. This is just a brief slide saying that there are biorelevant dissolution apparatus out there. The TNO-TIM apparatus there's a picture of it here. Apologies, it's not the best quality, but they actually have separate compartments to mimic the stomach, the small intestine, and the team there have done some work correlating in vivo data to the TIM model in paediatrics. And I've put a reference there for people who are more interested in that. I'm just going to move on a little bit now and talk about when you bring the biopharmaceutics tools together, so the solubility, dissolution and permeability, we come to the biopharmaceutics classification system, shortened to BCS, and that's a way of categorising drugs into one of 4 boxes dependent on their solubility and permeability. So a highly permeable drug, as I said before, there's these limits as to greater than 90% absorbed, and they're the chemical limits there. And the solubility for drugs is the highest dose strength soluble in 250 millilitres across the relevant pH range, and there's always some debate about the composition of these fluids. So that's really the way that you categorise drugs. And BCS1, so something that's highly soluble and highly permeable, the theory is that its absorption is less likely to be influenced by the formulation. So you can have a bio-waiver, minimise clinical testing when you change formulation. And this is quite important for paediatrics. because you may change formulation, do a study in adults, and then how does that back extrapolate to children? And this is an area you need to be a little bit concerned about. I'm going to highlight that in the next slide. I'm really going to focus on talking about the solubility here. And on the previous slide, I talked about the highest dose strength being soluble in 250 millilitres. And then we talked about that volume being smaller in children to account for their smaller anatomy and physiology. And this is just a slide coming from Japan, where they also use a smaller volume. They use a volume of 150 millilitres. So they're saying that the highest dose strength has to be soluble in 150 millilitres. But they also use lower doses compared to in the US, Great Britain and Spain. So this slide is saying that in Japanese populations, where you amend the dose in line with the volume, you actually don't get too many drugs changing BCF class. And this slide is quite important because it's a similar principle to what we're doing in children. Obviously children have a lower dose and a lower volume, and it's whether that change in both dose and volume affects the BCF or whether they're out of sync. So to look at this, I collected data, and I just combined two references, a WHO Essential Medicines list and also some paediatric formulation data from an FDA website. It was just to make sure there was easy access to the solubility, permeability data I needed for drugs that are used in paediatric populations. And this was the spread that I got. There were 56 drugs. They're nicely spread between the 4 BCF classes there. This chart just divides them by permeability on the y-axis and dose number on the x-axis, so solubility. And you can see I've got a good spread. And I also looked at the formulations used for the different products. The reason there's more than 100% is because quite often they're available in more than one formulation. And as you'd expect, for the BCF1, you get many more of these liquid solutions and tablets because it's easier to have different formulations for something of that type. This chart here is quite busy, but it's looking at the same data when we change the dose number, so we change the solubility, the dose soluble in the 25 millilitres. I'm going to go to the next slide because it's a little clearer. And this is really comparing those same drugs that we looked at for the adults, and now I'm comparing them to the neonate. So the adult data is still there in the blue. You can see they're nicely distributed between the 4 BCF classes. But when we put the neonate dose and divide that by 25 millilitres, we can see the solubility of these drugs becomes much poorer. So the dose numbers become higher, which means that a lot more of the drugs become poorly soluble, and the poorly soluble categories are the BCF 2 and 4. And this is quite interesting and quite important when we're manipulating formulations to see what effect that might have on the pharmacokinetics in a child of that age. These slides here have done the same as for the neonates, but for data for infants and children. And the real point that we're trying to make from this is here that the BCF4 drugs become much more significant in children's and paediatric populations. Therefore, they're much more sensitive to changes in formulation and how that might affect the pharmacokinetics. So I was looking at bioequivalence studies within paediatric formulations to see, again, to sort of bring home the importance of understanding the biopharmaceutics in children and adults. They identified 45 studies from a literature search that were conducted. As you can see, many of them are conducted in the adult population from the pie chart on the left, rather than the paediatric, and that's in line with the current regulations and guidance. What's interesting from the pie chart on the top right is that just over half showed equivalence between the paediatric and the adult formulation, and there was big differences in those that were higher and lower for the paediatric. Sometimes it's not too surprising because if you're making an oral liquid, you've made sure the drug's dissolved, so you might expect it to be have a rapid onset of action and be slightly better available because it hasn't got to go through the dissolution phase. So that's maybe not such a shock that there's some paediatric formulations that are higher, but those that were lower was certainly interesting. And the chart at the bottom just shows the different formulations used. Unfortunately, there wasn't enough data for me to go through and work out whether the bioequivalent ones were related to the formulations, because as I say, there's only 45 studies identified. But it'd be really good if we could mine more data to really bring any trends to the forefront. So this is about conducting bioequivalent studies, and this comes back from a conversation I had at the recent EU PFI meeting about, so we know there might be differences, we conduct them in adults, but should we be making a dose adjustment? So if we're looking at this dose to volume ratio, would it be more sensible to do a bioequivalent study in adults at a higher dose to bring out these differences, or should we not actually do it the same way? And the protocol, what volume should we use of the product? Otherwise we could ask an adult to take it with a reduced volume. And these are just interesting points that I thought I'd share. I don't have the answers, but I thought they were quite interesting ways to think about how we might design these studies going forward. I'm going to talk about food effects, sort of the remainder of this, because biopharmaceutics also covers differences in food effects. Obviously, food affects the absorption of drugs by interactions with the transporters, changes in the gastrointestinal composition, transit, etc. So I've looked at the food effect on paediatric formulations. About half of them had no food effect, and that they were bioequivalent with and without food. And several had a negative food effect, in that the exposure was reduced in the presence of food. And then I compared the food effects found on paediatric formulations that were found in paediatric populations and in adult data. So the graph on the left is about the formulations, the chart on the right is about products, where there's paediatric products tested in both the paediatric population and in an adult population. And again, what's interesting is where there's a food effect seen in one population, it's not always found in the other. So that's suggesting that the food effect isn't directly extrapolatable between adults and children. Some of these might be to do with the meal composition. So children eat somewhat different diets to adults. And here's just some examples where drugs and their availability have been affected by diet. So the studies there with etoposide, where the bioavailability was unchanged, it was taken with toast, but it was hugely increased when there was butter on the toast due to the fat solubility. And again, gluteoflorbin unchanged by certain meals, and the lipid meal changed it to a large extent. And finally, examples of diet for studies conducted outside of the Western population, where puri and dal gave a different effect to lentil rice cakes and chutney. The actual composition of the meal can affect the extent of increase or decrease in bioavailability seen, which is obviously important for clinical efficacy. This is just a little table to sort of talk about meal composition and the reasons why the different compositions affect it. There's also there's effects on the drug. So they may solubilize the drug if it's a fatty meal, but there's also effects on the physiology in terms of gastric emptying. So here you can see that there's changes to gastric pH, binding, you can stimulate bile and pancreatic secretions. There's all sorts of different effects that food can have on the gastric physiology and intestinal transit ties, and the impact that could have on drugs and their absorption profiles. So it's quite important that you understand this in terms of drugs taken with food. So I looked at the meal composition used in paediatric studies, and there was quite different meals used. Within food effect studies done in adult populations, there's an FDA standardised breakfast, and this is quite a very high fat meal. It's not a typical representative breakfast, but it's a standardised meal that's thought to bring about the worst case effects on bioavailability of the drug. So I think it's important to think about how we extrapolate that to children and have a standardised meal that is appropriate for all populations. So the different things that have been used are things like applesauce, yogurt, infant formula, obviously for children who haven't yet been weaned, milk and biscuits and cereal. And it's important to think about how this could influence the bioavailability of drug. The chart on the right is just about apple juice consumption. In that, and I'm not convinced that it's the actual issues, but you can just see there that the amount of apple juice drunk on a mg per cube basis vastly decreases once you get out of childhood. And again, milk consumption. Children tend to drink a lot more milk than adults, and it's just about thinking about something that represents their diet. There's a trend towards putting additional calcium in a lot of foods over in the States, and calcium is known to bind to certain drugs. So we need to think about global trends in diet, pH, typical meals, and how that can influence drug absorption, and make sure we understand the effects. It's particularly important for children rather than adults, where quite often in the home there's evidence that there's been studies done that look at how patients, their parents and carers manipulate medicines to make them more palatable to children, and often that does involve mixing with food. A second thing I wanted to just highlight here was something about eating patterns here. The amount of times one eats per day. Obviously, young children, babies are often fed eight or so times a day, whereas as an adult we conventionally think of people having 3 meals a day. However, this chart shows that actually there's quite a lot more eating occasions during the day, and it's about how often these and how they constitute a meal, because of the not only the gastric emptying and the effects on bioavailability, but how that matches their drug intake, or medicine intake I should say. This is just to do with meal volumes, and this is to do with what's the amount of food necessary to initiate the fed state, and then it's actually a very small volume in adults. It can be as little as 2 grams of fat in adults. So when we talk about feeding often and these eating occasions, how many induce the fed state and how many don't, and it's really to do with how much and what it is that you're eating. So neonates, have a small volume of feed, but it's often highly calorific, again because they're going through a growth phase and they need those calories to grow. And it's interesting that it doesn't correlate with weight. I did look at the extrapolation from neonates to school children, and the FDA breakfast has a volume of just over 500 millilitres. But if you do directly correlate the child's feed, it would be well over 2 litres worth of food, which is quite grotesque. And anyone who looked at the FDA breakfast composition eating much more than that would also be quite unpleasant. So this again is just something about food effects here in terms of differences. It's about the volume of food and the impact on the gastric emptying. It's the volume relative to the size of the stomach as to when that empties, the pH effect, the fat, and everything really. There's quite a lot to think about in food and food effect consequences. And it's to do with having the appropriate biopharmaceutical tools to really understand and probe these interactions. Just going to move on to paediatric physiologically based pharmacokinetic models. These are software tools that bring together pretty much everything I've spoken about from the biopharmaceutic element. They bring together physical chemical properties of the drug, information about the absorption of drugs in adult populations, as well as the paediatric physiology into a virtual population to allow you to predict pharmacokinetics in a virtual clinical trial. And these have been quite popular. They're very, very well used in adult populations. And I've just put 3 examples of providers of such software on the right. We've got SimFit, GastroPlus, and PKSim. So this is another element of biopharmaceutics where we collect data and where that data is used and how it's used. Just to put in a couple of slides about the regulatory status. This is a slide that I've directly taken from an FDA presentation, talking about how often these models are used in publications and also in regulatory applications. There's quite a lot of use now of these models in regulatory applications for paediatrics. My next slide, again, was data I've taken from a previous presentation showing about the top 5 uses of modelling and simulation. So using this PBPK software for paediatrics and paediatric drug development. And sometimes it's about analysis of pharmacokinetic data or simulations to guide a pharmacokinetic study to make sure you're sampling at the appropriate times. You can use it for dose selection and also to try and correlate the same, so you're getting the same exposure in a child compared to an adult. And also you can do some absorption modelling. So it's showing that there's a way forward using the data that we've got to try and predict how things perform. I've come to these conclusions by going through the different paediatric biopharmaceutics tools and where they're used and how they're used. And really, it's not a satisfactory conclusion that's going to say we've answered everything, but these are conclusions that were identified in those 2 key papers I put at the very beginning of the presentation by the US PSI and also the EU PSI, saying that we've got critical knowledge gaps where we really don't have enough data to extrapolate what we know that happens in adult populations to paediatric populations and thereby predict a pharmacokinetic profile. And the key areas are listed there. So it's about understanding the gastrointestinal tract and the ontogeny of that, as well as the liver and kidney, the permeability, fraction absorbed, and the methods that we're using to predict how things perform in paediatric populations, all the dissolution and solubility, what's the media, how do we measure those. And again, we need some more clinical data to make sure that we're validating some of these models that we've got. So I'm happy to take any questions, and apologies for the blocking out of the web system. I don't know whether it was our computers at this end, but apologies for that.
Well, Hannah, thank you very much. And don't worry about being blocked out. I've had a couple of messages suggesting that several people had some problems with connections during the webinar, not just you. But thank you for an excellent presentation, particularly as you had to pick it up on 2 occasions when you were blocked out. Hannah, we've got a number of questions coming in which I'm going to pick up in just a moment, but if I could use the chair's privilege and just ask you 2 questions. One is, it seems to me that you're saying that we should be rethinking the BCS classification for children of different ages. Is that am I getting the correct message there?
Yes, definitely. It's also been that's also been suggested by the US Paediatric Formulation Initiative, and I know they've got a work stream working on how they do that. So yeah.
Okay. My second question really is about the effects of food on bioavailability. It's common, we believe, for carers to mix medicines with food to administer them to children. Even though they may be palatable for one child, they may not be acceptable to the next. And so it's probable that mixing with food is much more common than we actually have data on. And my question really is about medicines where in adult studies we know that bioavailability is not affected by food. So if you were to recommend that medicine for a child were to be mixed with food, would it be necessary to do bioavailability studies to establish the fact that mixing with food does not affect bioavailability? Or for a drug where food does not present bioavailability problems in adults, is it reasonable to extrapolate?
At the moment, the regulatory sort of guidance seems to say that you do a food effect in adults, and whatever you find can be extrapolated to children. I don't know that that's a valid extrapolation, and I think there needs to be a lot more in-vitro tools that can help you predict this, because you cannot possibly do food effect studies on all the types of food in all the populations. So I think we need better tools to evaluate it. But currently, the suggestions are that you do your studies in an adult, and what happens in adults also happens in children with food.
And that becomes almost another question, but I won't ask it you now. I'll just comment that it becomes difficult to know what's reasonable to expect companies in developing medicines to do when they are testing medicines with different foods. Applesauce and yogurt appear to be the popular ones, but of course they will not fit all countries and cultures. Okay, so we have a question from our colleague Etienne Oju at UNICEF, and she's reminding us that scoring of adult solid oral dosage forms for use by children is something that is recommended frequently, for example, with antiretrovirals. So, Hannah, based on your work, can we conclude that scoring of tablets and splitting of tablets would be okay generally for children of 2 years of age and over?
It's really to do with the dose and the drug that's in the tablet. It depends. If it was, say, a poorly soluble drug within that tablet, you may expect that its dissolution be very much slower in children compared to adults. So you may find for a poorly soluble drug in a tablet, it's not appropriate, yet for a highly soluble drug in a tablet, it may be appropriate. And that's kind of the boundary we need to look at, is the solubility and finding those volumes to say which ones it would or wouldn't work with.
Okay, and Etienne has also asked another question about dissolution media and the apparatus and the timings and so on that you've mentioned. Is there guidance to the industry already on this issue? Does it form part of regulatory assessment just now?
No, there's no regulatory guidance at the moment on this issue, and that's recognised by industry as an issue in its own right, because I think each company is sort of looking at what they should be doing and how to get there, and there's such limited clinical data out there, it's hard to get a really good in vitro and vivo correlation, and that's an area where I know there's a lot of work going on, but there's no answers at the moment.
Okay, and we have a question about bioequivalence. So when you mention higher or lower bioequivalence between adult and pediatrics, can you comment how much lower or higher and which age groups?
Off the top of my head, I can't comment on individual ones. Where I said there was a difference, I used the regulatory guidances where there's difference in bioequivalence, which means that the confidence intervals are outside the 80 to 125% range. So if you put the adult product, the Cmax values, the peak plasma concentration, and the area end of the curve, if the paediatric product was the mean and the confidence intervals are outside of it, 80 to 125% of that range, it was inequivalent. Does that make sense? So it's outside of that range, but I can't tell you how much it is off the top of my head. But there's 45 studies identified, and I'm hoping to put that into a paper where it be fully referenced so someone could go through and look them up.
Well, I think that's the way we should deal with that, Hannah. I'm just sorry. I'm trying to multitask, which for a man is not easy, as you'll know. So Anne-Marie Korkinen, one of our paediatric committee colleagues, would you have suggestions for how to approach risk assessment for differences in bioavailability in children versus adults by means of in vitro methods, since we don't know, for example, what pH to use for gastric pH? So how could you risk assess, and what methodologies might you then go on and use?
This is where there's a lot of ongoing research that hasn't read out as yet. I've got suggestions as to what would be useful to do, from my opinion, based on the literature and the suggestions that the EU PFI group put into our paper, but there is yet to be any definitive boundaries, if you like, or definitive industry standard methodologies. And that's something, again, that's been acknowledged within the UK as an unmet need. And I've been working together with a wider group of colleagues to try and address this and get some industry standard methodologies for in vitro prediction of biopharmaceutic data for paediatric formulations. Again, it's about pooling all the data that we've got to give a big enough sample to evaluate and then validate techniques. And it's that area that's limited.
Well, thank you, Hannah. I'm sure that Anne-Marie can email you directly for a conversation on that area. We're coming towards the end of our session. First of all, I want to thank Hannah for her presentation, particularly managing her presentation with the interruptions that she had because of technical problems. So Hannah, thank you for a fascinating talk. I'm certainly pleased that we've recorded this, because it seems to me that I now need to go away and really study what you've said. It's difficult for a non-expert such as myself to take it all in 45 minutes. So that's my task over the next week, is to do that. But thank you very much for the presentation. Hannah will answer questions on the GRIP blog during the next 4 weeks. So if, like me, you're going to listen again to the webinar and questions crop up, then please go to the blog and you can ask relevant questions there, and we'll get some answers to you. I'm reminded that we sent you a pre-webinar poll just to find out what your knowledge was about these in vitro techniques and their application in pediatrics. We will also send you a post-webinar assessment. So when we send you the link, please fill that in so that we can get some understanding of how your knowledge may have increased. Mine will have gone from 0 to quite a lot in a short space of time. And finally, it just remains for me to let you know that our 6th webinar is coming up early in 2014. This is a very relevant webinar, because many of you will know that the final version of the the final version of the guideline on pediatric drug development has now been published by the European Medicines Agency and is available on their website. And Piotr Kozorowicz, who's one of the scientific administrators in the quality section at the European Medicines Agency, is going to talk to us about quality and the pediatric investigation plan, and no doubt will be referring to the development guideline at the same time. So thank you all for participating. My apologies for the technical problems. We will talk to our technical colleagues and see if we can iron them out for next time. And thank you once again to Hannah, and that's the close of our session. Thank you very much.